Thank you everyone for joining us for today's webinar. My name is Tom Ewing and I am with RTI International's Forensic Technology Center of Excellence. Before we get started, let me go over how you can view and interact with today's webinar. The first thing I would like to draw your attention to is our chat, which is located on the left-hand side of your screen, marked chat. This is going to be the best way to communicate with us if you have any questions regarding today's material. Feel free to use the chat to ask any topical questions you have during the presentation. All questions pertaining to the material will be retained until the Q&A session that will be held at the end of the webinar. This chat is also the best way to let us know if you experience any technical issues. If you do experience any issues, please feel free to type them into the chat and either myself or one of our customer service representatives will make sure you get taken care of. In order to have the best viewing and listening experience, we recommend using the latest versions of the following internet browsers: Microsoft Edge, Google Chrome, Apple Safari, and Mozilla Firefox. All your audio for today's webinar is going to be streaming through your computer speakers. If for some reason you happen to lose your live audio or live video feed, the first thing we would like you to do is to simply refresh your web browser and then hit the play button in the middle of your live feed window. This should refresh your connection with both the live audio and video feed for today's event. If for some reason this does not work, or you experience any other technical issues, please make sure to let us know in the chat pod. You can also utilize the toll-free number in our email address that are located directly above the live feed window to contact us with any customer service issues. We always like to find out a little more about our audience, so if you could take a few moments to type in your location into the chat, this will give us some great feedback about where everyone is joining from and ensure that your chat function is working properly. For today's webinar, we have Dr. Heather Jordan from Mississippi State University presenting the utility of microbes in forensic science. Without further delay, here's Dr. Jordan.
Hi, everyone, and thank you for joining the webinar today, where I will be talking about the utility of microbes in forensic science. My name is Dr. Heather Jordan, and I'm Associate Professor from Mississippi State University in the Department of Biological Sciences. So I like to give an outline of what I'll be covering, and you'll see this slide several times throughout the presentation so that you have a roadmap of the organization of the talk. So I'll start by giving a brief introduction, then discuss both classical and culture-independent microbiological methods that are used in forensic science, then highlight some published studies of microbes in several forensic areas, and then end with the needs that must be met for chains of custody and then some sampling methodology that goes along with it. So hopefully you'll come away from this webinar with a basic understanding of the difference between classical and culture-independent microbiological methods, with knowledge of data associated with forensic microbiology research, and with an understanding of some requirements for chain of custody with going from the field to the laboratory. So first, on to the brief introduction. So the one thing you might be asking yourself, what is forensic microbiology and how does it differ from molecular epidemiology? Well, certainly molecular epidemiology has been used for years to trace outbreaks of microbial diseases. And actually many of the methods used in molecular epidemiology are the same as what's used in forensic microbiology. But the difference here is that forensic microbiology data has to hold up not only to critical observation by the scientific community, but also to judges and juries. So forensic microbiology is thus a marriage between the scientific method and the criminal justice system. And now forensic microbiology is a relatively new field. And in fact, the push for the utility of microbes in forensic science increased really after the anthrax attacks in the United States in 2001. Now, since then, the use of microbes has been considered pioneering in forensic science. And the ability now to study microbes at the genomic level using some of these cutting-edge next-gen sequencing techniques have led to the power to both accurately and consistently use microbiological data and these methods in forensics. So it's thought that microbes and the human microbiome, which is defined as the collection of microbes in and on an individual, are expected to provide insights into the origin of biocrimes, into cause and time since death, and also to give evidence for other crimes such as medical malpractice and sexual assault. And some recent studies by our group have also shown that postmortem microbiomes, or those microbes associated with deceased individuals, can be predictive of the antemortem health state and can also be useful for tracking baseline antibiotic resistance genes. And I'll talk about some of those studies towards the end of the talk. So now I want to get started with a brief comparison of classical culture-based microbiological methods and culture-independent methods that we use in forensic microbiology, along with some pros and cons of each. First, getting into classical microbiology, where traditional culture-based microbiology, biology, as the name implies, allows for measuring growth and metabolism in pure culture. So these microbes can be grown in liquid media or on a semi-solid surface. And once a microbe has been obtained or cultured in pure culture, it can be analyzed phenotypically or visibly for growth requirements or patterns of growth, as you can see in some of these figures here. One could also look for antibiotic susceptibility, even toxin production. And many other utilities as well. So these characteristics can be valuable for identifying unknown microbes, and the methods have been used for years to advance our knowledge of microbes as a whole. But there are some restrictions that limit our ability to analyze some organisms in this way. For instance, out of all the thousands of microbes, thousands and thousands of microbes out there, only 1 to 2% of all the microbes are culturable under laboratory conditions. And those that are cultured, some of them may be fastidious with some specific growth requirements, such as, the need for blood or serum or some other things. Some may also have long growth times where it may take weeks for them to grow, which is obviously not ideal in a forensic context. So then we can utilize also culture-independent methods. And even those microbes that could be obtained in pure culture can also be studied using molecular methods such as PCR, or polymerase chain reaction, or even sequencing. And sequencing allows detection of targeted genes as well as entire genomes of specific organisms. But I mentioned that cultured organisms can be sequenced, but most microbes outside of the laboratory grow not just a single species, but as a mixed microbial community with thousands of microbes together. Analyzing a microbial community using these next-gen sequencing methods is a powerful method then to study genetic material recovered from these mixed populations. And using these methods, one can study microbes all the way down to the strain level from both living and dead organisms that may even be unculturable. So obviously, these methods have greatly advanced the field of microbiology and also for use in forensics. So what happens first after a sample is taken is usually that microbial DNA is isolated. Then a specific microbial gene may be amplified using PCR. A common PCR target is the 16S ribosomal RNA subunit found in prokaryotic organisms whose structure is shown here on the bottom left. The 18S ribosomal RNA subunit can also be used when targeting eukaryotic organisms. And this can be followed by sequencing of that amplified targeted unit. So the sequences can then be compared to known sequences that are archived in online databases. And you can see in this figure, this table on the top right here in the graph, that the known sequences in databases have vastly increased over time, as well as the accuracy of the curation within these databases. Now, other molecular methods include shotgun metagenomic sequencing, where DNA from the entire community is sheared and directly sequenced, or transcriptomics that determines the presence and abundance of expressed genes, proteomics for proteins, and also strain typing that identifies bacteria at the strain level. And this was particularly important for source tracking in the case where the Bacillus anthracis strain from the US anthrax attacks was traced to the likely origin lab. Now, there are advantages and disadvantages to both the culture dependent and independent methods. For instance, culture is relatively inexpensive. It allows to distinguish between living and dead cells, and it's also specific. But it may have low sensitivity, and as I mentioned already, growth may take a long time, even if the organism itself is even culturable. Molecular microbiology, on the other hand, is sensitive and specific. It can distinguish between strains depending on the target and usually has a fast turnaround time, but it can be very expensive depending on the method. It doesn't usually distinguish between viable and non viable cells, and it also requires the isolation of DNA or RNA or proteins. And these methods all require specific training and aseptic technique as well. But even after sequencing with this rapid turnaround, target specificity, there can be multiple reasons why an organism can still be misidentified. For instance, microbes are promiscuous and they exchange DNA with each other, and they have the potential to exchange, for instance, the genetic forensic marker that is being tested that could lead to false positives. Also, sometimes pathogenic and non-pathogenic strains can have the same sequences. And the identity of an organism can only be as good as the database accuracy. And though I mentioned that there has been an increase in the accuracy and curation, these databases are still not infallible. And sometimes new methods are just not yet validated. So obviously, the quality of information is an incredibly important aspect of using the data as evidence in a criminal case, and validation with precision and accuracy is incredibly important. But overall, both methods, despite their limitations, are valuable and have been used in several subfields in forensics. So now I want to get into the meat of the talk and get into research areas where these methods and analyses have been implemented, starting first with investigations into cause of death. And as I'm sure all of you all are aware, one of the first objectives when a person dies is to determine the cause and manner of death, to determine whether an individual died as a result of natural causes or by drowning or disease or intent to harm, such as by poisoning or biological attacks. So first, talking about drowning, microbes in the lungs and other organs due to inhalation and circulation during life, along with pathological signs suggestive of drowning, can be used in some cases and may be able to provide evidence for drowning, along with evidence for the location of drowning as well. So an individual that is drowning is inhaling these microbes into his lungs and their circulation during life. Microscopic phytoplankton called diatoms are the most common microbes used to establish drowning, and a figure up here on your top right are showing some diatoms there. And these diatoms are typically present in natural environments, and they can be compared from the samples collected from the individual to those diatoms in the local aquatic environment to give valuable conclusions regarding the possible place of drowning, and are sometimes actually the only valid approach in determining the time of death. But fecal coliforms have also been studied as possible indicator organisms for death by drowning, and there's some pictures of those on your top right there as well. These organisms are smaller than diatoms, so presumably they'd be able to migrate into the bloodstream faster. And they're also found in seawater and freshwater and are present inside the gastrointestinal tract, but are not naturally found in the blood. So then migration into the bloodstream would be an indicator of drowning. Studies have also shown that these are specific enough, these fecal coliforms, to confirm drowning in seawater and freshwater without yielding any false positives. Now, additionally, microbiological investigation is an important part of the autopsy process for identifying infection and may have significant implications for public health in documenting patterns of infection, managing case contacts, identifying novel infections, which we can all appreciate in these days of COVID, or bioterrorism threats, and then also to contribute to outbreak control. Additionally, the diagnosis of some infections, for instance, fungal infection, as shown in this figure here, as an example, may be missed or difficult to achieve during life, and so may be only diagnosed during the postmortem. And finally, a number of sudden unexplained deaths in infancy, or SUDI, cases have been associated to infectious diseases as well. For instance, one group conducted a retrospective review of 507 autopsies that were conducted between 1996 and 2005. And this study found Staph aureus and E. coli significantly more frequent in infants with unexplained death, and more so in the lungs. And that's what's shown in this graph here on the bottom right, where the lungs and the spleen are the number of organisms per sample are shown in the lungs and spleen, and the blue bars are the unexplained deaths. And you can see that there's a higher proportion of these particular organisms in the lungs and spleen, suggesting that these bacteria could be associated. Now, another study that I'm not showing on here investigated microbiological evidence in organs and fluids of 121 SUDI infants that were less than 2 years old. And they showed that cultures were positive for pathogens in 49% of the cases. And there were also cultures from non-pathogenic microbes in 73% of those cases. Now, they concluded from the study that infectious disease could lead to unexpected deaths due to bacterial pneumonia with a high proportion of gram-positive bacteria in these cases. So this just shows, just again, a thumbnail of the utility of microbes in determining cause of death. Now, microbiological evidence has also been useful in cases of sexual assault. So I want to highlight some research into the utility of microbes in this area. Microbiological evidence in sexual assault cases may come from body swab or fluid specimens, as well as bed sheets or blankets, clothing, hair, or even fibers. And these samples can be analyzed using PCR tests or maybe culture or other point of care tests. And these tests are used for the detection of sexually transmitted pathogens or for the detection of trace evidence. And there have been some studies that have shown that bacterial communities, not just single microbes, but bacterial communities themselves can be used as microbial signatures to trace their involvement in sexual assault cases. And actually, one study evaluated the merit of microbial metagenomic data of human hairs as evidence, so determining the value of human hair microbiomes. So the study analyzed sequences from DNA extracts that were obtained from human scalp and pubic hairs collected from 7 male or female individuals over 3 time points. Now, it's important for me to tell you that one of the males and one of the females was in a relationship with the other. So this figure here on the bottom left shows some of the sequencing results, where each dot represents microbial sequences in a single sample. Now, the different colors represent the different sample types. So if you take a look, for instance, in panel B, the green and blue dots are male and female scalp hair microbiome sequences. The female scalp hair is blue, male scalp hair is green. The closer the dots are to each other, the more similar the microbiome sequences are. So you can see that the blue and green dots are all mixed together, showing that the microbial sequences of the scalp hair really can't be differentiated between males and females in this study, showing that they really have limited value to particularly identify individuals. But moving over to panel A here, the orange dots represent male pubic hair microbiomes and the red dots represent female pubic hair microbiomes. You can see a separation of the men versus women microbiomes and clustering within each sample. So the orange dots from the male pubic hair are clustering sort of together, as are overall the female pubic hairs as well, except in one instance. So if you look at the red dots and the yellow ellipse, these represent taxa of the female partner of the couple at T0 and then the second time point. While the orange dots that are encircled by blue represent the taxa from the male partner at T0 and the second time point. The lilac circle encloses one red dot, which is a female taxa at the third time point, and one orange dot representing the taxa from the male microbiome at the third time point. So microbiomes from the male and the female at that particular time point that are encircled in this lilac are more similar to each other than to their other previous time points. So it was actually revealed that the couple had engaged in sexual intercourse 18 hours prior to the collection of hair samples at this third time point. And interestingly, both individuals had also showered in the interim period. So the authors had speculated that cross-transference of bacteria during intercourse may account for the variation in the taxa observed. And this was even detected 18 hours and after a shower. Also in this study, the authors also identified signature taxa indigenous to female pubic hair that can aid in discrimination between individuals. And that's what's shown in these two figures on the right. So this is lactobacillus that is found specifically in vaginal samples from women. Another study that recently came out was from penile and vaginal swabs, showing that the authors had identified unique microbial compositions in both the penile and the vaginal microbiomes that also, they differed from each other, but they also differed from skin microbiomes. Again, showing forensic potential of microbial metagenomic analyses. So overall, both of these pilot studies are promising, and they suggest that microbiome analysis can be of evidentiary value in sexual assault cases, along with other evidence, or potentially even when other associative evidence is not present. But of course, there are some limitations that have to be considered. These are pilot studies, but other considerations are that, for instance, male pubic hair is thought to be seldom transferred. There's also been an increase in the use of prophylactics in sexual assault cases that would limit sample collection. And then, of course, there should be consideration for when there are multiple sexual partners, and there may be legal and ethical challenges associated with interpretation of data and also confirmation. So now I want to go a little bit further and get into forensic microbiology data for trace evidence. Trace evidence, as you all know, is physical evidence that can be transferred from contact between objects. It can be used to identify the location of decedents and witnesses, and is an essential component for including or excluding witnesses and unlocking circumstances leading to death. It's not surprising then that understanding the microbiological associations between people and their surroundings has become a powerful tool for providing evidentiary data, especially with respect to location. So with trace evidence, this evidence is carefully collected, traditionally in the form of hair fibers from the victim's or suspect's clothing or from the crime scene. But bacterial communities or fungi or even fungal spores can also be picked up by any object, and it's also similar to other evidentiary considerations, such as those from hair or fiber. And these can serve as a proxy of environment from where they came. It's not conclusive evidence, but it is definitely an important consideration. And one of the first cases of microbes as trace evidence was investigation of a young woman's body that was found dumped in a patch of stinging nettles. So there were spores from 2 fungal species that were associated specifically with these stinging nettles that were found both at the crime scene and in the suspect's car. And that was thus a link between the suspect and the place where the body was dumped. Now, when confronted with this evidence, the suspect actually confessed. Now, the human microbiome has also been applied in forensics for trace evidence. And this is because there is a high degree of inter and intradiversity between individuals and body locations. So you can look at this figure here of the human microbiome on the right. And this is a figure showing pie charts of different types of bacteria associating with different body locations. And it's not important for you to be able to read or see the different types of bacteria, but just understand that the different pie charts are associated with different body locations, and the different colors within each of the pie charts are different types of bacteria. So it's showing that bacteria have specific niches that they associated with body location, and also between individuals as well. Now, the human microbiome is also relatively stable and can be recovered just hours after body washing. And the majority of taxa fall into 4 phyla as shown here. There is relatively high diversity at the species level, but as I already mentioned, there's high interpersonal variation, making it possible for use in forensic identification of individuals. Actually, there was a study by Franzosa et al. In 2005 in PNAS that showed individuals could be uniquely identified among populations of hundreds of people based solely on their microbiomes. And interestingly, greater than 80% of the individuals could still be uniquely identified up to a year later based on their gut microbiomes. There's also been shown to be influence of interpersonal relationships, and it's been shown that microbes, just like other trace evidence, can be transferred from contact between objects, and we saw that in the previous study of sexual assault. And human contact can transfer millions of microbial cells, again, making it possible to elucidate personal interactions between individuals. So for instance, microbes of hands, feet, and noses were found to be similar from individuals in the same household, and they became even more similar if there was physical interaction. And also transfer can occur even with short-term contact, and studies have shown skin microbiomes are more similar after competition. And personal effects are also individualized, and personal effects can become more similar when people inhabit a space. So there was a study conducted by Lax et al. In 2015, and I'm showing some of the data here, where 2 participants sampled floors, shoes, and cell phones over a 2-day period. And some of that data is shown in this figure on the left here. Now, another 89 people took individual samples at 3 different conferences, and some of those data are shown on the right. So looking at the data on the left first, results showed that surface types yielded different communities. So these are results of the 2 participants, as I said, and this is looking at shoe and phone sample similarity. And they are colored, the different colors represent surface and person. So you can see person 1 and person 2 in blue are associating, have their associated microbes with phones. And then there's the shoe microbiomes of persons 1 and 2 in red. And then floor is shown in this yellow-orange color. So the shoe and the phone sample microbial communities clustered, first of all, by person, and then also by surface type. And also what came out from this study was the impact of floor to shoe community was significant in that the different sites maintained a significantly different floor microbial community, which in turn shaped the microbial assemblage structures associated with the shoe samples. So you can see that here where you've got person 1 shoe associated with the person 1 floor that they came into contact. The same with the person 2 floor as well. So without going into too much detail, let's take a look at panels D and F over on the right-hand side. So these show volunteers at the 3 different conferences, those 89 people that took samples. One of the conferences was in Vancouver, showed in orange dots, one in Washington shown in the green dots, and one in California shown as the pink dots. So the D panel is showing the microbial communities associated with the shoes at the 3 locations. And F is for the phone microbial communities at the 3 different locations. And you can see clustering of the dominant microbial communities associated with the shoes and the phone at each location. So this is beneficial. These data are really beneficial and useful, not only for geo-tracking, as we can see that we can identify location and surface types, but this may also have merit for tracking diseases in a hospital setting. For instance, by monitoring any longitudinal dynamics of microbial communities in the hospital, or maybe tracking influential variables about the building or the occupants, and even potentially for personalized medicine. But also, these types of data could provide clues to a person's lifestyle or maybe ethnicity or diet, and even regular associations between individuals. All right, so now I'm going to switch gears a little bit more and talk about postmortem interval, and then get into some microbial data involving understanding time since death, starting first with studies of soil microbial communities. So time of death, again, as you all know, I'm preaching to the choir here, is an extremely critical part of the evidence. It can exclude or include witnesses and suspects. So if the time in question isn't known, there are a number of medical scientific techniques that are used to determine it. Forensic analysis of body decomposition for estimating the postmortem interval or time since death are mainly derived from taphonomy, or those changes a body undergoes following death, as one can see the progression here in these images. Or in some cases, scientists will use organisms such as insects that have colonized a set of human remains over decomposition time as well. So microbes play a significant role in decomposition, and microbial presence, abundance, and transmigration, or the movement of these microbes, have been studied for use to estimate postmortem interval by analyzing, first of all, the grave soil, but also cadaver microbiota. And I'll focus on the soil microbe communities here. So there was a study by Carter and group in 2007 that showed that there is what's called a cadaver island, where cadaver decomposition results in release of ammonia-rich nutrients into the soil that then alter pH and nutrient content that will then stimulate microbial growth. But then also other factors play into the microbiome, such as animal scavengers and insects that increase microbial activity, also add to the microbial community by bringing and taking away their own microbiomes as well. So another study showed that exogenous soil communities impact the rate of decomposition. So this is a figure by Schoenenlauber et al. In 2014 that showed that exogenous soil communities impact the rate of decomposition where cadavers reached in this study active decay faster in untreated soil, as shown in the bottom figure, than in sterilized soil. So what you're seeing, let's look at the bottom here. You have a decomposition time on the bottom, and then you see the different stages of decomposition on the right-hand side. And the bottom graph is for untreated soil communities, and the top is for sterilized soil. And what you see is that the cadavers that were placed in the untreated soil, so not killed, there were microbes present, reached active decay much faster in fewer days than if placed on sterilized soil. So there's another study, not shown here, that soil microbes can become conditioned by previous episodes of decomposition, where soil previously containing decomposing muscle tissue promoted decomposition of fresh tissue faster. And the authors attributed that to specific bacteria that were able to quickly utilize the cadavers. Also, there have been other studies that I won't go into detail on, but that have shown that there are a number of factors that are affecting soil-associated microbial communities, including the influence of temperature or moisture, soil texture and type, whether a body has been buried, the type of season. Their decomposition occurs 3 times slower in winter than in summer in one study. And also, as one might expect, cadaver-associated microbial communities impact soil-associated microbial communities and decomposition as well. So we'll get into some of those studies of how cadavers impact microbial decomposition and potential use for postmortem interval. And we'll start with those using animal surrogates to better understand this microbial succession and the impact on decomposition. So there have been several studies conducted that have revealed complex dynamics associated with decomposing animals in both terrestrial and aquatic environments using mice, swine, or fish carrion. So for instance, Melvin et al. In 1984 used culture-based methods to investigate postmortem microbial transmigration through the small intestine of mice, and he found that transmigration of species is temperature dependent, and that there was also a shift from aerobic organisms to anaerobic organisms. Metcalf et al. Used a large number of mouse replicates and also targeted metagenomic sequencing, and showed that decomposition was different across body sites, that soil microbial communities were different from the cadaver microbial communities, and that communities changed in a similar successive manner in each replicate as decomposition progressed, with specific taxa, specific microbial taxa, dominating at different decomposition times and stages. And Peckel et al. Used swine and 16S metagenomic sequencing and also showed transmigration of species is temperature dependent, depending on season and insect activity. They also showed a shift from aerobic organisms to anaerobic, and also showed specific taxa dominating at different decomposition times and stages. And then there are a number of authors that I won't go into have shown the utility of fish, swine, and even whales in determining the postmortem submersion interval. So how long a body has been in the water. Now, we also conducted a study, my collaborators and I, with an NIJ funded study, where we inoculated immunocompetent mice with fluorescently labeled Staph aureus through the nose, because Staph aureus is a facultative anaerobe and is a normal inhabitant of the nose in many individuals. Or we inoculated orally with Clostridium perfringens, that is an anaerobic inhabitant of the gut. So we chose two different types of microbes that could live in different oxygen conditions and also in different areas of the body. So we surface sterilized a subset of cadavers following sacrifice. So I want to say we inoculated these mice while they were alive. Then we sacrificed them and then surface sterilized a subset of these to determine the impact of the external skin community versus the internal community. And we also wanted to investigate transmigration from an initial site to other organs immediately following death. We had a particular interest with colonization of previously sterile sites. So we also used in this study microbial total RNA to measure microbial succession over time and also the gene expression. So this is a figure showing our Staph aureus study, showing evidence of increased Staph aureus transmigration occurring in the non-surface sterilized mice over decomposition time. Staph aureus was at the highest detectable concentrations in the nasopharyngeal area within the first 24 hours. You can see these fluorescent areas where we inoculated and the mice had inhaled up into the nasopharyngeal area. There was increased visible evidence of transmigration from 3 days to 14 days in these sample sets. So we also detected our tagged fluorescently labeled Staph aureus using qPCR in all of our organs as early as 1 hour post-sacrifice across our sampled organs. So we also measured microbial community succession across organ and time and found that bacterial diversity was dependent on organ and also postmortem interval. We also detected similar trends as to other animal models. So these are figures of graphs showing relative abundance of microbes at the family level across organs and time. So just to orient you, this is a relative abundance of microbes in the stomach. And if you look from the bottom to the top, the bottom three are microbiomes associated with mice at 3 hours, and then 9 hours, 26 hours, 74 hours, and 170 hours in the stomach. The same for, goes for the intestines, the heart, and the bone marrow. So what we showed was that there was this succession of microbes across decomposition time. And again, we found things similar to other studies where there was this movement from aerobic microbes to anaerobic. And again, we also were excited to find actually that we could detect microbial presence in this particular case as early as 3 hours post-mortem. So we also identified from this study, I mentioned we isolated total RNA, we identified significantly differentially expressed genes in the heart. And in our other study, we also identified some significant expressed genes in the bone marrow using 2 different software packages for measuring differential expression. So in this case, heart bacterial gene function increased with genes for amino acid metabolism, as shown here, stress response, sporulation, and motility. So we were also from this study able to show that total RNA analysis provides both structural and functional data, so who's there and what they're doing, from a single sample in a cost-effective manner. And the specific genes that were upregulated, we were able to differentiate here early versus late time points and middle versus late time points. Now that's about as far as our precision was. We weren't able to differentiate early versus middle time points in this study. But the genes that were shared among that were found were shared among many microbes. So it wasn't just one microbe with a single gene. It was multiple microbes sharing this same upregulation of the same gene. And this could be useful in designing a field-based analysis kit in the future. So now I'm going to be talking about the utility of microbes in determining postmortem interval with some studies involving human bodies. So the first two studies I'm highlighting here in this slide utilize bodies that were donated to body farms. So in the first study, as shown a figure on the left here, Hyde and colleagues sampled 2 bodies every 2 days for 2 weeks, then every 4 days for another 2 weeks. They took samples from several skin sites, also from the mouth and the rectum. And they showed initial dominance of proteobacteria in early sampling, then a shift to Firmicutes phylum at later sampling times. They also showed that shifts in microbial community corresponded to major taphonomic events. And they also show what's shown in this figure here is that microbial communities became more similar to each other over decomposition time. So if you look towards the end here, days since placement, the further from placement, the closer the different microbiome samples were to each other, indicating that they became more similar. And this was for both bodies. So the second study was by De Bruyn and Houther, who repeatedly and sterilely sampled gut material from the cecum of 4 cadavers that were placed at the University of Tennessee Anthropology Research Center in summer 2011. These individuals had died of natural causes and been refrigerated prior to placement for 3 days. During sampling, the authors also observed extensive insect activity. So they used 16S targeted sequencing, and they found that taxon richness increased, as if you look in the figure here on the right, these again are relative abundance tables of 3 of the 4 individuals that were placed. And so they found that taxon richness, or the number of different species types, did increase over decomposition time, but there was a decrease in the relative abundance of the different taxa of each of them. So they were also, from this study, able to identify potential indicator taxa associated with postmortem interval, one of which was a microbial genus associated with insects, this Ignationaria. And they also found differences between the 3 analyzed individuals as well. So those were studies from body farms. This next bit of data is from a large-scale study by our group led by Dr. Jennifer Peckel that identified postmortem microbiomes from 188 human bodies that were received into the Wayne County Medical Examiner's Office. So samples were taken from these bodies during routine autopsy. And in this study, the swabs were collected from the eyes, the nose, the mouth, and the rectum during routine autopsy. So if you look first in panel A and the figure on the left, again, the shapes and the colors represent different sample types here. As you can see, eyes, nose, ears, mouth, and rectum. And from these, we found that microbial communities were dependent on anatomical location. So you can see clustering within microbiomes of the rectum, general clustering of the mouth, ears, etc. We also found that the number of differing taxa decreased as decomposition progressed, and that's shown in panel B here. So this is mean 16S diversity, and you can see on the left-hand side samples from bodies that had postmortem intervals less than 48 hours versus more than 48 hours, and you do see this decrease in differing taxa as decomposition went from less than 48 hours to more than 48 hours. And then panel C, without going into detail, shows that there was reduced microbial community variation within the first 2 days since death, suggesting increased microbial community stability in the first 2 days after death compared to later communities. So what that means is that the microbial communities, the postmortem microbial communities in this study up to 48 hours postmortem were more similar to ante-mortem microbiomes and were stable. So the top figure on the right, where it shows B here, shows some of the identified taxa, and many were commonly identified in living hosts, such as Staph or Strep. And we also found, as shown in the bottom figure, a positive association between predicted genes for microbial cell motility and time since death. So overall, all of these studies using human bodies adds to the growing literature of postmortem microbiome communities and microbial succession that contributes to a better understanding of the decomposition process and its potential utility in forensic science. So now I am going to switch gears a little bit and talk about how postmortem microbiomes can aid in predicting the living condition and also be used for antibiotic resistance source tracking. So that same study by Peckel that I just went through, Peckel et al., from those postmortem microbiomes of 188 bodies, I mentioned that they showed stability within the first 48 hours postmortem. Also in that study, we showed evidence that microbial biodiversity is a predictor of antemortem host health condition. So for instance, heart disease. So you can see in the figure on the right here where microbial diversity decreases in this case here, as does specific measured taxa associated in cases with heart disease compared to cases without heart disease. So this is showing decreased diversity. And then specific taxa here, the blue bars show no heart disease and the red bars show heart disease. And so it's showing this decrease in diversity of these specific taxa. On the other hand, individuals whose death resulted from violent circumstances, as shown in C and D, had an increased microbial diversity. And there was some slight decrease in specific taxa, but overall there was this increase in diversity. Now, a follow-up study using those data showed that beta dispersion, defined as this time-varying spread, this dispersion of microbial communities in samples. So beta dispersion, along with case demographic data, could distinguish among manner and cause of death. And this table shows that here. And this is especially true in differentiating between cardiovascular disease and drug-related deaths, which were correctly classified in around 80% of cases, as shown here. Now, understanding that postmortem microbiomes are stable within 48 hours and also understanding that they may be predictors of the antemortem health state, we utilized human body samples collected during autopsy and in this case within a 72-hour period after death for biomonitoring and as a surrogate model for antibiotic resistance genes presence and abundance within the living human population. So we invasively collected and characterized microbial communities from several different body locations collected from 39 recently deceased individuals. Of these, 17 were Caucasian, 21 were African American, 13 of these were female, and 25 were male. Individuals were classified into discrete age groups, as shown here. They were also scored as normal, overweight, obese, or not applicable based on their body mass index, if known. 8 individuals died of natural causes, or their manner of death was undisclosed, while 8 of the remaining 12 were ruled homicides. So with this, we used whole genome shotgun metagenomic sequencing for some samples. And then we also used commercially bought qPCR antibiotic resistance gene arrays for some other samples. So our whole genome shotgun sequencing results are shown in the top right here. And it's showing different antibiotic classes. And then on the right-hand side, these are the different body IDs, so different individuals that were sampled. So we identified 95 genes detected multiple times across individuals with resistance against 12 different drug classes as shown here, or a multi-drug efflux pump. And so, again, this is a heat map, and different colors represent the number of genes associated with resistance against each antibiotic class. So blue is none, all the way up to this brick red color that is greater than 40. So the maximum number of antibiotic resistance genes in a single individual was 70, but interestingly, 4 individuals had no antibiotic resistance genes detected at all. Now, using our qPCR arrays as shown on the bottom here, we detected 24 genes across all the individuals sampled, with 18 detected multiple times, and resistance in this case against 6 antibiotic classes or multi-drug efflux pumps. So in this case, macrolides and beta-lactam resistance was highly detected, and actually going back up to this whole genome shotgun sequencing, tetracycline and multi-drug and also macrolides were among the highest gene types that were also detected. So this is a table showing genes detected across all the different bodies. And genes found in the highest abundance were ERM-B and MEF-A, which are associated with macrolide, or ERM-B is a macrolide genes associated with macrolide resistance. But notably, genes were found for resistance to carbapenims, to vancomycin, and methicillin A. I'm including this OXA group here. 5 were detected for the OXA group. And carbapenem is used to treat multi-drug resistant bacteria. So finding genes for resistance to carbapenem was a little bit alarming. We also found MEK-A, which is a gene responsible for methicillin-resistant Staph aureus in 55% of our individuals. So methicillin-resistant Staph aureus is a huge problem in hospital settings. VAN-B was also found in 20% of individuals, and this is associated with vancomycin-resistant enterococci and vancomycin resistance as well. And interestingly, vancomycin is used to also treat methicillin-resistant Staph aureus. So that finding both of those within our cohort was not pleasant. But beyond that, in 3 of our individuals, we actually found both MEK-A and VAN-B genes in these individuals at the same time. So this is extremely important to know for population surveillance. This study also allowed access to sampling areas on and in the bodies that otherwise would not be routinely accessible in a living individual, and underscores the value of autopsy collected samples for a more holistic antibiotic resistance gene surveillance approach. So we were able to sample multiple areas of the body invasively. You all wouldn't want me to invasively collect samples from your nose or gouge you in the eye to collect these samples. But this was a small scale study, and a more robust study would be needed along with a cost assessment to determine its value for population surveillance. So I want to just mention other subfields of forensic science. I know time is passing quickly, such as medical malpractice and some other non-traditional forensics. So though I don't have time to go into specific studies, I do want to mention that there have been several research studies and studies in cases involving microorganisms in medical malpractice, where there was a need to identify whether there was intentional infection or negligence, in some cases misdiagnosis, and even in tracking nosocomial infections. And it's been shown that these types of investigations could and have provided litigible insights into chains of transmission to help to control nosocomial infection. And there have been some other studies and use in cases that have proven or refuted plaintiffs' claims of a hospital-acquired infection due to malpractice. And finally, microbial data and evidence has been used to identify instances of environmental contamination, foodborne illness, plant pathogens, and even some cases of animal cruelty. So even though I didn't have time to go through it, I would encourage you to look up those studies to learn a little bit more about them. So finally, I wanted to finish up by talking about the needs that must be met for chains of custody and sampling methodology. Proper documentation and validation is most important for trial, obviously, and is at the crux of every investigation. So evidence that was located, for instance, at the beginning of a case may potentially become critical later on. And so the chain of custody and evidence, if properly documented, will make it easier to locate the necessary information as the case progresses. As with any evidence, microbial evidence should be authenticated before it can be deemed admissible in court to prove that collection processes were sound and void of tampering, and to maintain a documented chain of custody. But there are some special considerations for microbial evidence that differ from other routine samples at crime scenes, such as the history of the specimen and the environmental context. Is it a swab collected from a body? Is it soil? How complex will the microbial community be? Are you looking for microbial communities versus a single organism? Also, the physical and chemical composition as well. Pathogenicity, whether there is potential for high toxicity, and what the biosafety level may need to be. Also, what type of personal protective equipment is necessary for the person collecting the samples and also other members of the entire team at the scene. The type of downstream analyses, meaning is viability important? Do you want to culture it versus running it for isolation of DNA and RNA and those type of downstream analyses? And with that, what type of transport procedures or preservatives may be necessary? And there's a need for aseptic technique across no matter the sample type, not only to protect yourself, but also to protect the sample. We all have microbes associated with us and on us, and we actually shed our own microbial cloud. So it's important to collect samples in such a way using protective equipment where our own microbiomes don't contaminate the sample. And of course, standardization of methodology will be important to have precision and accuracy and methods and comparable results that can be used in court and to validate the methodology. So forensic investigation should include a well-developed evidence collection and handling plan overall prior to collection. All right. So that wraps it up. I hope you all have enjoyed the presentation. This was a very dense presentation, I know, but yet, even though it was dense, it's certainly not exhaustive. I highlighted only a few studies in each of the subfields. I teach forensic microbiology at Mississippi State and usually take an entire semester to cover all of this content. But overall, I hope you've come away from the presentation with obtaining these learning objectives of understanding the difference between classical and culture-independent methods used in forensics, knowledge of current research in several subfields, and knowledge of the needs that need to be met for chains of custody and for forensic microbiology to, have its day in court, if you will. And with that, I thank you for your attention and will entertain any questions. Thank you very much.
Thank you, Dr. Jordan, for a fantastic and informative presentation. Let's go ahead and get right into some questions from the audience while we still have some time. First one I have here is there an increase for specific microbes in sexually transmitted diseases? And if condoms were used, would microbes still show?
Yeah, that's a really good question. To my knowledge, there hasn't been an increase in specific microbes associated with sexually transmitted diseases in relation to sexual assault, although there are some increases in STDs without sexual assault, but that's another topic. But as far as condoms, yeah, that is a limitation, particularly when trying to get trace evidence. Certainly, that does limit detection potentially of the microbiomes from the penile microbiome, and it could also limit transfer of hair, but it wouldn't limit it 100%, right? Based on how the condom is worn. So there could still be transfer of hairs where microbiomes of those hairs could still be potentially useful, but it definitely is a limitation in that it does limit the number and types of collection in those cases.
Another question I have here, how do you think the labeled Staph aureus transmigrated so quickly through the mice in your study? And apologies for butchering the scientific name of that.
That is not a problem at all. I actually get that question frequently, and it's because, I don't know 100%, but these microbes microbial mechanisms for decomposition begin, pretty much at death, and these microbes modulate their gene expression, presumably to be able to survive in this changing environment, right? So they would upregulate certain genes while downregulating others, and although staph doesn't have a flagella or pili that would allow it to, be purely modal or able to move, it can still passively move through liquid media, through different mechanisms. But also, if you look at the genes that we found to be upregulated, cell motility was also upregulated from that early to late time point, and also the middle to late time point. So presumably there were other microbes whose cell motility was upregulated. And interestingly, it's found that though Staph aureus doesn't have these flagella, it can move through the media, but it can also it can also hitchhike on other modal bacteria. So those that do have flagella, it's been shown to be able to hitchhike on those. So that's possibly how it could get through. Now, one other reason could be a potential limitation to the study where, in that study, the microbes were inoculated in the nose, and we had sedated the mice. And once they came to, they sniffed it up their nose, but it could be that they swallowed some as well, which could have allowed, more quick diffusion to other areas.
I think we have time for one more question I've got here. Is it possible to determine if two people had contributed to a sample rather than the sample just being single source?
Yeah, now that's been shown. I don't know in the context that I know, with skin samples, analyses of skin microbiome samples. Yes, you can, particularly when you're looking at like that study of cell phones, for instance, that was attributable to the individuals that were holding the cell phone. So the surface of it was traced back to the person, as was, there's another study of computer keys as well. And there was an interesting study in the home as well, where the different surfaces were swabbed, and it showed that, there's a similarity of microbiomes, but one of the people in the household left for travel, and there was a certain cohort of the microbiome samples that went away during travel. So yeah, there is this individuality, at least in the studies that I'm aware of, that do show contribution of a single individual versus multiples, where it can be teased apart.
Thank you. I appreciate it. I think that is it for our questions. So we'll go ahead and close out today's webinar. We would first like to thank our subject matter expert for taking the time out of their day to present the material for today's webinar. So thank you again, Dr. Jordan, for taking that time and providing us such a wonderful presentation.
My pleasure. You all have a good day.
We would also like to take this time to thank our audience for joining us today. These webinars could not be possible without your attendance and support. Before we leave today's webinar, we would like to ask you to participate in a short survey. You can locate that link back on the webinar's main page. Simply click the word survey, and that will bring up our quick customer satisfaction survey for today's event. Your feedback about our webinars is vital information that we use to provide you with the best webinar experience and adjust the content and the way we disseminate the information to our audience. One more thing to draw your attention to, you should see we are putting links in the chat to all our social media pages, Facebook, LinkedIn, and Twitter, our website, forensiccoe.org, and a link to our podcast episodes. Make sure you are following us on social media and subscribe to our newsletter to stay up to date with everything the FTCOE is doing in the forensic community. With that, I'd like to thank you one more time for attending, and we hope everyone has a great rest of your day.